Hi everyone, Dr. Hinkie here with a video lecture on Chapter 3, our chapter on the cell. So I'm not going to do a video lecture for Chapter 1. I think that was some interesting overview information that I think you probably all found very helpful. But I think that was pretty easy to read through. We're going to go into more depth on some of the important points later on in the semester. That really was just an introduction. But the cell, here we're going to introduce some information that is probably going to be new to a lot of you. Chapter 3 has a lot of information. It's assumed that the whole second part of the chapter, eukaryotic cells, you're quite familiar with, either from 101 or from an A&P class. I am going to do a second video that does a quick review of eukaryotic cells and then goes into the specific eukaryotes of microbes that are covered in Chapter 5. I'll post that second video this weekend. So we're going to go over the first part of Chapter 3 on prokaryotic cells. There's a lot of interesting information in here, and of course the cell structures are going to come up again and again. So the chapter starts with some history of the cells and discovery of cells. Our objectives in this first section are to explain the theory of spontaneous generation, and then to explain the role of Louis Pasteur in disproving spontaneous generation. We've looked at some of the historic figures. I'm very excited to read the enthusiasm that you're showing for learning some of this history. It is really fascinating when you think about trying to study microbes and bacteria pre-microscope and pre-tools that we have now to look at these organisms on a molecular level. So it's pretty ingenious, and as the discussion has shown, a lot of times these were more accidental discoveries or just someone's passion that they pursued. They were persistent in finding an answer. They had a question they wanted to know. Not necessarily that they were just these brilliant geniuses, but they were persistent. So I think that's one thing we see over and over again in science, is that persistence really pays off. So we'll take a little closer look at some of these historic figures that loom larger than life in our textbooks, but really were just living people. So pre-Louis Pasteur, people believed in spontaneous generation. So if you think about this in the before the microscope, before we could see on a microscopic level, what you could observe, what you could see was that, hey, there was nothing living here and now there's something living here. Frogs just suddenly appeared in water where there hadn't been frogs before. And maggots showed up on meat that was left behind or unspoiled food from nothing. These, mice would show up in the barn in a pile of rags. Where did they come from? Well, they came from cells, or they came from mama mouse who had baby mice. But if this wasn't actually observed, if you didn't see mom having baby mice, and mom usually kept those pretty hidden until they just showed up in your barn. Maggots just showed up on your meat because those fly eggs were invisible. So from an observational standpoint, it looked like life just spontaneously arose in these places. And so one of the first experiments that showed, well, no, maybe we need something else in this picture, was Francesco Redi's meat jar demonstration, where he put some meat in an open jar, and lo and behold, maggots. Well, the flies were flying in and out and laid their eggs. But when he sealed the jar, we didn't have any maggots. Well, nothing got in there. And if we covered the jar in cheesecloth, we'd find maggots on the top of the cheesecloth because the flies were attracted to that smell. He knew there was a food source there, and they would lay their eggs there. It would be on top, but not on the meat. So that suggested that living organisms were coming from other living organisms. But we still needed conclusive proof. And there were different experiments going on. Really, the conclusive, absolute, definitive, we disproved spontaneous generation, and developed the theory of biogenesis. So genesis is the creation, bio of life, that living things come from other living things. So Louis Pasteur did this very elegant experiment. A lot of these were when we look at them now, we say, oh, it's brilliant. And the reason they're brilliant is because they're really not complex. They're very simple, makes them elegant. These simple experiments where he filled a swan neck bottle. So these swan neck flasks, this looks like a swan's neck. He put broth in that. And this would prevent bacterial and fungal spores from entering. They could get to here, and then they'd sort of get trapped down in this bottom part of the swan's neck here. Have liquid, it would prevent things from coming in. So he boiled and sterilized that broth, and when it was cool, it remained free of contamination. But if we took off that neck, the part that was preventing anything from coming in, then it would get contaminated. It would get cloudy. This is something you look for in lab if you're working with nutrient broths, is cloudiness. And that cloudiness are all the bacteria that are growing in there. So this is a great quote. Life is a germ, and a germ is life. It's a living thing. Never will the doctrine of spontaneous generation recover from the mortal blow of this simple experiment. So that disproved spontaneous generation. That became a part of modern cell theory, is that all cells arise from other cells. So we're going to briefly look at the development of that cell theory and how that has evolved. Well, we tend to learn in textbooks at this level that the cell theory has 3 basic statements to it. There are really about 72. So if you think back to the scientific method, a theory is a predictive model that has been supported by extensive experimental testing. Numerous related hypotheses are tested, and the more evidence we gather supporting those hypotheses, the more we try to disprove them and we can't, the stronger our foundation comes. At some point we get enough information, we get a clear enough picture that we can build a model. It can be a predictive model that we can say, if I go out and look for this in the real world, here's what I'm going to see. And then we go into the real world and we see if we can find support or some way to disprove or discredit the theory. And the more we try and can't, the stronger it is. So we'll look how our cell theory, these 3 statements that, just remember, they're really 72 statements or more than that, quite a few to give us a complete picture because we know a lot about cells. Look at how those sort of support everything and how they have evolved up to close to present day with the endosymbiotic theory, which is still being expanded. We're still testing components of this. We're still proposing hypotheses and gathering evidence on this. And then we'll look at a few more people who have some incredible contributions to our cell theory and germ theory and things that we'll be studying. So cell theory, the idea of a cell, Robert Hooke, back in the late 1600s, so think about this, about Civil War time, was the first one to describe cells. And he looked through a microscope at cork. So cork is the bark of an oak tree, quercus. And this is what it looks like. And to him, what that reminded him of is in monasteries where the monks are in cells. They're all in rows. Think like a prison cell, very similar, all nice, neatly rowed, same sized squares. And plant cells tend to be larger than many animal cells and very regular in their shape and structure. And so that's what that reminded him of. And so he coined the term cell. Not 1865. I said Civil War. What date was I looking at here? Dyslexia. 200 years before the Civil War, we got the term cell. So some other contributors to this early idea of cells being the foundation of living organisms. Matthias Schleiden was a botanist who observed that all plants are made of cells. He looked under the microscope and saw this same structure everywhere he looked. Theodor Schwann was a physiologist who saw that animal cells have a lot of similarities to plant cells. So animal cells don't have a cell wall, so they don't have that always the same repeating structure and very similar rectangular shape that plant cells do. But he did see that there were a lot of similarities between these cells. In the 1800s, so now we're up to close to Civil War times. So just to put that into some historical context of what we knew when. So it's before turn of the century, before World War I. Robert Remak died in 1865, but mid-1800s, they proposed this extension of abiogenesis as part of the cell theory. All cells arise from cells. So initially what Pasteur had proposed is that living things come from other living things. This takes us down to a smaller level, to the single cellular level. How did we get to become a multicellular organism with 35 trillion cells? It was by cells reproducing and making more cells. That's through mitosis. You remember that from Bio 101. And from there, we continued to expand the modern cell theory to say that all cells only come from other cells. So we aren't going to get a cell from anything other than another cell. And cells are the fundamental units of all organisms. There's usually a third statement that goes with this. It says the cell is the basic unit of life. And that really goes hand in hand with cells are the fundamental unit of organisms. Because a cell by itself, a single cell, can be a living organism. But we are not 35 trillion living organisms. We're one living organism made of 35 trillion eukaryotic cells. So those two sort of go hand in hand to account for both multicellular organisms and single-celled organisms. If we removed any one of our cells, yes, it would still be living. It could carry on all the functions of life, but only for an indefinite amount of time. We couldn't keep that alive indefinitely because it's part of a multicellular structure. It needs its related cells, whereas a single-celled organism can perform all the functions of life by itself. So that's why we usually have those two statements together, is just to clarify that point. So in the 150 years since then, continued to study cells, continued to learn more about them as new tools and new methods became available to study them. As we've been able to look more and more closely, we have more powerful microscopes. Now we have molecular mechanisms that let us look even more closely at cells to continue to refine and develop and add to this theory. There are still questions we don't know. Where did the first cell come from? There are a few competing hypotheses about this, and if you're interested, you can look those up if you'd like to see what's being proposed and the evidence. There's evidence for the various hypotheses that are out there. So the primordial soup tends to probably have the most evidence. Its main competitor is, I can't remember what the name of it is. It's basically that the first cell came from outer space, which leads to the question, where did that cell come from? So doesn't really answer the question. And only the first cell on Earth. I want to say spermatogenesis. Panspermia, that's what the name of that one is. All right. So anyway, where we've evolved now, we have learned quite a bit about cells, and both bacterial and eukaryotic cells like our own. One of the big questions about cells was how did we get to these big complex cells in eukaryotes from this single cell, the original cells on the planet that are simple life form. Everything started with a single cell. How did we get this great diversity? How did we get from that to these larger, more complex cells, and then to multicellular organisms? In the 1970s, let's get started a little bit earlier than that, but a researcher named Lynn Margulies, so maybe in the 60s, it was before the 70s, in the 60s, Lynn Margulies proposed the endosymbiotic theory. And what she looked at was looking closely at the components of a eukaryotic cell. She said, look at that. In animals, the mitochondria look an awful lot like bacteria. And they're about the same size as many bacteria. And then looking at plant cells, she said, hey, look at that. The chloroplasts look a lot like bacteria and are about the same size as bacteria. The function of a mitochondria is to produce energy. Well, living organisms acquire materials, use them to get energy, and use that energy to build structures. Plants use the energy from sun. The chloroplasts use energy from sun to build food. And there are prokaryotes that are photosynthetic that look an awful lot like chloroplasts. Lynn was pretty much dismissed at the time because she was a female working in biology in the, 50s, 60s, 70s. She got a nice little pat on the head and was dismissed. But she persisted. And as technology caught up with her and she was able to look more and more closely, it turns out that the chloroplast and the mitochondria have a double membrane, which is exactly what would happen if one cell ingested another cell. You would end up with an outer membrane and an inner membrane, that outer membrane being part of the endocytosis process, packaging that cell that we just ingested inside a little membrane-bound transport vesicle. And then, so that was some evidence that, that's how those could have gotten in there. Then we got better and better technology. And she was able to extract ribosomes and analyze them. And lo and behold, eukaryotic ribosomes are structurally different. The ribosomes found in animal cells and plant cells are structurally different than the ribosomes that are found in prokaryotes. The mitochondria and the chloroplasts have ribosomes, but they're not eukaryotic ribosomes, even though these are organelles found in eukaryotes. They're prokaryotic ribosomes. And then she was actually able to extract DNA. And it turns out that mitochondria and chloroplasts have their very own DNA, suggesting they were their own individuals. Now, some of the things that have contributed and expanded our understanding of this that we found come through those mitochondrial DNA. When we are born, in the original egg cell and sperm cell that come together, the mitochondria are found in the egg. The sperm only contributes DNA. So our first mitochondria comes from our mother. So our first mitochondria is identical to our mother's. Subsequent mitochondria come from that first mitochondria dividing. And lo and behold, the mitochondria divide separate and apart from the rest of the cell. But we look at that DNA, and the DNA in our mitochondria is identical to that in our mother's mitochondria. Doesn't matter if you're male or female, Our mitochondria came from our mother, and hers came from her mother. And so we can track ancestry back through mitochondrial DNA very, very, very accurately, because that mitochondria, the only change over time is just this background mutation level. So mitochondrial DNA, tracking that through the maternal lines in eukaryotes, where it would come through the maternal egg, has given us even more evidence that Lynn Margulies was onto something with the endosymbiotic theory. And at the time, it was a hypothesis. Now there is more than sufficient evidence that it is accepted throughout the scientific world, throughout biology. And sort of that process was, there was an original prokaryotic cell that engulfed an external cell, an aerobic one in the case of mitochondria, a photosynthetic one for the chloroplasts. Rather than digesting it for food, something happened, some glitch. We'll look at that as we go through the term. And it didn't digest it, but instead it kept it inside and found, hey, look at that. This mitochondria, or this bacteria, is pumping out this energy that I can use. What a good benefit. So whatever that glitch was genetic. Passed it on to its offspring, who also engulfed and failed to digest some bacteria that produced energy for it, and on over millions of generations, until mitochondria became first a symbiont with the cell that engulfed it, and then eventually a complete part of it. So mitochondria is no longer a separate free-living organism. I can't extract my mitochondria and have it go on. They are now intricately bound together as part of the same cell. That was over millions of generations. But the mitochondria retains its own separate DNA. When the cell is about to divide, the nucleus for the eukaryotic cell sends a signal to the mitochondria, and the mitochondria divides, so that it can get ready to split and have one mitochondria in each daughter cell. They can also divide during the regular life of the cell, because some cells need more than one mitochondria if they need lots of energy, like muscle cells. And then plant cells, similar process going on with photosynthetic cells. So as I said, this doesn't end our theory. We continue to investigate, look at new things. In the late 80s, Lynn Margulies said, hey, I'm looking at prokaryotic or bacterial flagella, and boy, they look a lot like sporulum, which is one of our shapes of bacteria. And so she started looking at the structures and seeing if maybe that flagella, the bacteria that have flagella, if maybe that's an endosymbiont, if that's a relationship that had evolved over time. That's still up for debate. All right, so that's our cell theory. And I see science is progressive. We continue to study and learn and expand and improve our knowledge. So we're still studying cells, even though we know a lot about them. There's still a lot that we don't know. Another theory that we'll be interested in is germ theory. The germ theory of disease states that disease may result from microbial infection. I am not going to play the videos in this, but when you go through the PowerPoint, please do click on the links for the videos and watch these videos. Ignaz Semmelweis was an obstetrician, and what he noticed, his observation was that, physicians would go, from room to room in the hospital. They would perform an autopsy, and then they'd go into an examining room and examine a patient, or go to the delivery room and deliver a child, and without washing their hands or without changing their clothes or anything in between. And so he proposed hand washing. He said, gee, why don't y'all wash your hands in between? And kind of like some people today who don't believe in wearing masks or washing hands or social distancing, people rejected this idea of hand washing. Bah, it's ridiculous. How is that possibly going to prevent disease? And after his death, lo and behold, people started washing their hands, or physicians started washing their hands, and childbed fever, the mortality rate in maternity wards significantly decreased. He also compared that infants born by midwives at home were more likely to survive at this time. And midwives were hand washing, were very big on promoting cleanliness during childbirth. So now we celebrate him as the first proponent of hand washing as a way to prevent the spread of disease. But it did not happen in his lifetime. Another researcher who contributed to our studies of germ theory and the spread of the disease is Jon Snow. Everybody can remember Jon Snow from Game of Thrones. This is not that Jon Snow, but a British physician in the 1800s who tracked down, he mapped cases of cholera. As cholera was spreading, he mapped where cases were popping up. And at this time, cholera was thought to be this poor person's disease. It tended to happen in crowded cities, in around more densely packed areas in the slums. And so people would say, oh, that just affects poor people. As he tracked it, though, when wealthier people in the suburbs and further from the city started to get sick, then people started to get interested in what could possibly be the cause. He tracked this and mapped it and found that he could find where sort of how the disease was going to spread by seeing there are more cases here, fewer cases out here, fewer out here as I get further away. And if I track back to where the most cases are, there's a common water source. And that common water source was where people were contracting the cholera. That was the source of the cholera outbreak. And so he's really considered the father of epidemiology. Epidemiology is, now everyone is much more familiar with it, but similar to forensics in that it is trying to track and solve questions about the spread of disease. So he's the father of epidemiologists, and he did this by mapping where people got sick when. Louis Pasteur, we've already talked about. So disproved spontaneous generation. And he also discovered that microbes are responsible for food spoilage. They're also responsible for fermentation. And based on that, he proposed that, hey, if they can spoil food, if they can ferment liquids, beverages, then they could probably cause some problems in our body. They might be responsible for infection. So he had proposed that, but didn't have any way to demonstrate that. And as we were introduced to in Chapter 1, Robert Koch is the first person who came along and gave definitive proof. He established a causative relationship, not just correlation, but causative relationship between a specific microbe and a specific disease. And he did this with an experiment where we'll get into this in the book a little bit later, but he had a series of mice that were all infected, all had the same disease, and he isolated bacteria from them. And then he isolated what was the common bacteria in the sick mice that the healthy mice did not have. He injected that into healthy mice. They all developed the same symptoms, same disease, and died. And then he was able to isolate that same bacteria afterward from those mice. So showing definitively that these bacteria present with this disease cause this disease in healthy animals. So he was able to identify the causative agent, and he looked at a number of different bacteria and diseases. So Koch's postulates are sort of the 4 steps that are needed to clearly link this pathogen with this disease. And we'll look more closely at those in a chapter coming up. Joseph Lister, we see Listerine. He is a surgeon that was the first to develop aseptic techniques in medical settings aimed at reducing microbes. He went beyond just washing hands with soap and water between visits to different rooms to actually disinfecting hands and air with carbolic acid. This is phenol, and phenol is sort of our number one, can be very toxic, but it's our number one cleanser that we rate all disinfectants against phenol. And we'll look, when we talk about disinfectants and control of growth, we'll look at what phenol is and how it works. But Joseph Lister is the first one who began disinfecting using aseptic technique. So this gives us our timeline, and if we look sort of here back in 1546, we have our very first proposal of germ theory, and yet it took almost 350 years till turn of the century before someone was able to specifically link cause and effect from bacteria and infections. So ongoing, progressive, little by little, as we learn more and more, we can build on that. I think it's quite an interesting history. A lot of things that we take for granted now, like, hand washing, aseptic technique when you're performing surgery. Those were revolutionary at the time. All right, so now let's get into our cell-specific structure. So if we look at classification, we have three domains. We have the bacteria and the archaea, which are single-celled simple organisms with no membrane-bound organelles. So our domain is bacteria, archaea. These are both single cells, so they're both very similar. They were both in the same domain until fairly recently, when we got molecular techniques that showed that, yes, they have similar ribosomes, but our ribosomes come in two parts, and the archaea ribosome is half similar to bacteria and half similar, has a lot of similarities to eukaryotes. They also have some different cell wall structures, so they are significantly different enough to be separated out. It's also was thought that bacteria can cause disease in humans. Archaea, we don't know. They've never been known to cause disease in humans. Maybe they can. For quite a while, it was just sort of a given that they don't because we've never seen it. But now some researchers are suggesting that, yeah, maybe they can. And then the eukaryotes is what we're familiar with, are bigger complex cells that consist of the kingdoms Protista, which are single-celled organisms, and algae, which algae fall into that, even though we have macroalgae that are very huge, like kelp. The vast majority of algae are phytoplankton, or single-celled organisms. And they don't fall quite into the category of plants. They don't have plant-like structures. The kingdom of fungi and plants, and then what we know, animals. All of these kingdoms have more similar cells. They have nuclei, and they have membrane-bound organelles. Their cells are much more similar in structure and function than our bacteria, which are different in our archaea. So we are going to focus on our bacteria. Just a quick review. We look at cells, the different types of cells. All cells, the characteristics that all cells have, whether they're prokaryote, eukaryote, plant, animal, fungus, bacteria, archaea, they all have DNA. They all have RNA, and they all have proteins. A lot of textbooks just say DNA because RNA comes from DNA and proteins come from DNA and RNA. But they do have all of those. All cells have those. You would never find a cell that doesn't have these. They all have cytoplasm, which is the aqueous solution inside the cell in which everything floats, and that's where all of our resources, all of our the chemicals that we need, the ions that we need, everything that we take in from eating, sort of floating around in here to take place in chemical reactions. The cell membrane, sometimes referred to as the plasma membrane, is our outer wrapping. And both bacteria and eukaryotes have plasma membranes. Plants have plasma membranes, animals, all cells have a plasma membrane. This is the structure that separates inside the cell from outside the cell. Oop, that doesn't belong there. All cells don't have membrane-bound organelles. That's my bad. I meant to cut and paste that there and I got it in the wrong place. They do not have membrane-bound organelles. Sorry. Cross that out right now so that we don't get confused by that. And then I'll change the slide in the D2L. Ribosomes. They all have ribosomes. Ribosomes are an organelle. They are a small structure with a specific function inside cells. The function of ribosomes is that they are the site of protein synthesis. All cells have ribosomes. They are an organelle, but they don't have a membrane around them. So that's why we say that membrane-bound organelles are only found in eukaryotes. So our prokaryotes, if we compare, they have all of these things. They're very simple. They don't have a lot of complexity. They're very small. They have no membrane-bound organelles. All bacterial prokaryotes have a cell wall. For the ones that we're interested in, for our bacteria, they have a cell wall made of peptidoglycan. So that's what we're going to focus on is peptidoglycan. There are two groups of bacteria that we're not going to talk about that don't have a cell wall, but we're really not going to talk about them other than mycoplasmas and L-form bacteria do not have a cell wall. They are the weird, bizarre exceptions. All the bacteria we're interested in have a cell wall that's made of peptidoglycan. And all prokaryotes exist as single-celled organisms. There are no multicellular prokaryotes. Eukaryotes, the cells are significantly larger, 10 to 100 times larger than prokaryotic cells. They all have a nucleus that is a membrane-bound structure that contains the DNA. They are complex. They have compartments. These compartments are membrane-bound organelles. That's where that membrane-bound organelle belongs. I found it. Oh, no, there it is. They have membrane-bound organelles, and that's what gives them compartmentalization. So they have endoplasmic reticulum. These are little separate rooms. They have Golgi apparatus. They have mitochondria. They have all these little separate rooms. So different functions can take place in different parts of the cell. I like to compare these to a one-room schoolhouse versus one of the buildings on Trident Tech campus. One-room schoolhouse, grading, teaching, discipline, administration, paperwork, everything, lunch, phys ed, everything takes place in the same room. Bacteria, everything takes place in the same room. In eukaryotes, I have separate compartments for separate functions. So different things are separated, so chemical reactions can be isolated and take place at a faster rate. There's less interference. Things have to move around, and that just makes for more efficiency and more ability to specialize, and that's given eukaryotes a great advantage. So we're going to focus on prokaryotic cells. Our objectives are to explain the distinguishing characteristics of prokaryotes, to describe common cell morphologies and arrangements, describe internal and external structures, and compare characteristics of our bacterial and archaeal cells. We've done that, really. Our bacteria and archaea differ. Archaea have some structures that are found more similar to eukaryotic cells, and they don't cause disease. These are sometimes called extremophiles because they're found in extreme environments. They can be methane producers. They are found in deep sea hydrothermal vents. They are found pretty much everywhere we say, oh, nothing could possibly live in that environment. In the hypersalinity of the Great Salt Lake, nothing could, oh yeah, archaea are found there. Nothing could be found at the Great pressures at the bottom of the sea. Oh yeah, archaea are found there. So they tend to live in very extreme environments. And that's about all we're going to talk about archaea. So onto our bacteria. We'll also describe structure and function of the plasma membrane, of the cell wall, and other external structures found on them. So this is our sample prokaryote. Of course, we'll see everything shown on here. These are all the different structures that we might find on a prokaryote. So this is a typical prokaryote. It's hypothetical, meaning you're probably not going to find one that has all of these structures. So we are going to look at the different structures. This is just an image to show all the possible structures our prokaryote could have, and one shape, the bacillus shape. So this is so that I can run through and talk about all of these different components: the plasma membrane, the cell wall, the capsule, the pilus, the flagellum, the fimbriae, cytoplasm, ribosome, the nucleoid region, the inclusions and plasmid. Those are all things that bacteria could have. What all the bacteria that we will talk about have is a plasma membrane, because all cells have a plasma membrane. Outside of that plasma membrane, our bacteria will have a cell wall made of peptidoglycan. All bacteria will have cytoplasm, the liquid matrix, the aqueous solution where everything the cell needs is sort of floating around and suspended inside here. Ribosomes, the site of protein synthesis. All cells have those, so all prokaryotes have those. The nucleoid region, we don't have a membrane surrounding our DNA and separating it from everything else that goes on in the cell. But wherever the DNA is concentrated, we tend to call that we say that's the nucleoid region. This is an aqueous solution. This could float all over the place. It could be anywhere. But DNA, all cells have DNA. So there it is. And all the rest of these structures, some bacteria have, some don't. And that's really how we identify a lot of bacteria. Which of these structures do they have? Which ones don't they have? And then biochemically, what do they do? What are they able to eat? What are they able to process? How can they use different resources? So that's how else we can differentiate them. But from physical appearance, this is what we're looking at. So if we look at the structure of our bacterial cell, all bacterial cells possess the cell membrane, the cytoplasm, ribosomes, and that chromosome, their DNA, a single circular chromosome. Believe it or not, that hodgepodge messed up thing of string, if we spread that all out, it would be one single circular chromosome. But it's floating in liquid, so it gets all tangled up. Most bacterial cells possess a cell wall. As I said, the exceptions are the mycoplasmas and the L-form bacteria, which we're really not going to talk about other than to mention they don't have a cell wall. And most bacteria have some sort of surface coating called a glycocalyx, but not all. So structures that are found in some prokaryotes include flagella. So flagella, you're probably familiar with that, are used for motility. Pili, they're sort of a long, looks like a flagella. It's really a long modified fimbriae. And the fimbriae are these thread-like structures that extend from the plasma membrane out through the cell wall that are used for adhesion. So these are all external appendages, different uses. Some have an outer membrane. So they have the plasma membrane and then the cell wall and then a membrane layer outside of that. Not all, but some. Plasmids, which plasmids are these little extra chromosomal bits of DNA. So here we said this is a single circular DNA. This is all the DNA, this is all the information that tells this bacteria how to survive, how to function, what to do when. Plasmids are these extra chromosomal bits of DNA. The bacteria doesn't need it to survive, to function, but it's just sort of like bonus DNA. A lot of times this is how bacteria exchange genetic information. They'll take a plasmid and copy it and say, hey, this gives me some really cool features. And they'll make a copy and share it with another bacteria. So plasmids are just some little extra bonus DNA. Inclusions are little clumps or clusters of materials, of resources, things that the bacteria needs that it wants to hold on to and store. They're kind of storage units. A lot of these are made of proteins, but they can be gases. They can be other things that they're just little storage units. Endospores are a specialized structure that some bacteria make that puts them into essentially hibernation, a dormant stage. And then intracellular membranes. This is just a few instances of this, not really common. So we'll look at each of these more closely. First, we'll look specifically at our bacteria, since our poster child bacteria here is a bacillus. Just make sure we know not all bacteria are bacilli. Bacilli or bacillus is singular, bacilli is plural, are rod-shaped. They can be very tiny rods. It's just that they are longer than they are wide. So it's an oval, it's not a circle. Sometimes under the microscope it can be hard to tell because they can be very tiny rods. Cocci, or the singular coccus, like streptococcus. Coccus is spherical. These are round bacteria. We can have helical or curved bacteria. And these we have vibrios. Vibrios are comma-shaped. So we have the Vibrio cholera that causes the causative agent of cholera. That's the shape of that bacteria. We can have spirillum, or spirilla in the plural, that are spiral. They're kind of helical shaped. They twist around. And spirochetes, which have more of a distinct curve. Spirochetes are rigid versus spirillum, which are flexible. And these move by axial filaments, so their motion is sort of like a corkscrew. We can also have arrangements. Not all bacteria have arrangements. Arrangement means how they line up one to another after they divide. Sometimes they divide and they just split apart in their individual cells. So you'll just look under the microscope and see lots of individual cells. Some bacteria species tend to stick together after they divide. Each cell is still functioning. If I split them up, it's not a big deal. They just continue to divide. But as they grow, they tend to stick together to get larger and larger masses. And these can be pretty distinct under the microscope. So the arrangement that I get, if I just have I can have a single coccus. I can have lots of single coccuses that are unconnected. I have some that when they undergo binary fission and split, one cell becomes two. Those two stay together. And then as these divide, we'll keep splitting up, but we'll stay in pairs. We use the prefix diplo to define that, diplococcus. We can also have diplobacilli. You might hear that pronounced diplo. British and English tend to have a lot of different pronunciations for these, so you'll hear both in the videos. But we can be diplo, two, di being two. We can divide again and end up as fours or tetrads. The two we're going to see more than anything with cocci. If they stay in clusters, they kind of look like clusters of grapes because they're round. We call that staphylo. So staphylococcus can be an adjective describing my bacterial arrangement and type. So staphylo, it's a cluster of cocci. I can also use that term Staphylococcus as the genus, so I have to know the difference. Am I talking about the genus Staphylococcus, in which case it would be written in italics with a capital S, or am I talking about this as an adjective describing the shape and arrangement of my cells? In case it would not be italicized and it would be lowercase Staphylococcus. If my cells stay in chains, stay close together in big long strips, I call it strepto. So streptococcus are chains of bacteria. And I remember that by saying streptobacilli, a long strip of bacillus. So those are my different arrangements. And sarcina are when I get little cubes, so 4 by 4. All right, if I look inside my cell, if I go inside, look under an scanning electron microscope, I would see this area that's a little more dense. I see this big long squiggle through it. I would define this as my nucleoid region. They said we were floating around in aqueous solution, the cytoplasm, so that's not always necessarily right in the middle. It can be all clustered up and clumped up on one end. Can be spread out, could be just about anywhere, but wherever I would find it when I'm looking under the microscope, that would be the nucleoid region. So it's the condensed area of DNA that's visible as not a separate entity, but I can identify this under the microscope. The DNA is circular, it's double-stranded, just like our chromosomes, except our chromosomes are in threads. And I have many of them. Here I'd just take one of those and circle it around to get my single bacterial chromosome. And then those plasmids, these little extra chromosomal pieces of DNA, they're small, also circular, double-stranded DNA, because that's what DNA is a double-stranded molecule. Bacteria can copy these separately and pass them on. I'm not keen on the word offspring here because they can make a copy, and when they undergo binary fission and divide into 2 cells, they can pass that on. But more commonly, they make copies of that and pass that on to other cells around them. So passing genes down from one cell to offspring to subsequent generations is horizontal gene, or is vertical gene transfer, going vertically down the generations. But plasmids are often copied and passed to peers in their cohort. So I'm going to pass that on to my friends in my same generation through horizontal gene transfer. These aren't essential to bacterial growth and metabolism, and oftentimes they code for things that we call virulence factors, things that actually help the bacteria to survive, and many of those things are also the things that cause problems, disease, and illness in us. So antibiotic resistance is often held on plasmids, not in the regular chromosome. Tolerance to toxic metals, the production of certain enzymes and toxins can also be directed by that plasmid. So you can see, hey, I have this cool little chromosome here, little bit of a chromosome that makes me resistant to this antibiotic. I'm going to make copies of it and share it with all my friends. So since we reproduce just through binary fission, we make a clone, an exact copy of ourselves. There's not a lot of genetic diversity other than through mutation. So this is one of the ways that bacteria have overcome that. To diversify is to be able to share beneficial genes. Let's see. These are used in genetic engineering. This is they're easy to manipulate, transfer from cell to cell, get them to carry specific genes, and we will talk about that later. Ribosomes are the site of protein synthesis. So all cells have ribosomes. In prokaryotes, we have a specific subunit of 30S is the size. The large subunit is 50S. When they come together, the total, it's actually not cumulative, it's overlap. This is refers to a molecular size, actually, and the complete chromosome together in the two parts we call a 70S chromosome in prokaryotes. Ribosomes are made out of proteins and RNA. The type of RNA they're made out of is ribosomal RNA, and we'll learn more about that in the genetics unit. Cytoprotein synthesis, this is where I'm going to build all my proteins, because proteins, especially the class of proteins known as enzymes, are responsible for all the chemical reactions that take place in cells and metabolism. This is the sum total of chemical reactions in a cell. Without enzymes, we would have no metabolism, so we would have no life. So these are pretty important. We're going to spend a lot of time looking at these and how they work. So protein synthesis, we're going to make, especially for bacteria, the enzymes that I need to function. Inclusions are little intracellular storage bodies. They vary in size, number, and what they hold. And bacteria can use those to sort of store resources, store food, when those are depleted in the environment around them. They can store sugar. They have gas vesicles in aquatic bacteria, which are really bubbles to help them float. They can store sulfur in little protein-bound packets and phosphate granules. So it's a way to package. They don't have membrane-bound organelles, but this is a way to package and retain materials. Endospores. So some bacteria are capable of producing endospores. And what an endospore is a dormant resting cell. So some gram-positive bacteria like Clostridium and Bacillus have the ability to do this. Not all cells do. But these cells would be normal cells just like we looked at, and they would be metabolically active. We call that a vegetative cell. But if the conditions around them became very adverse, very harsh conditions, all the food is gone, we're in a drought, extreme heat, pretty much any adverse environmental condition, the bacteria is able to form an endospore. So it's a protective structure that isolates the bacteria from the surrounding environment. It becomes dormant, is no longer metabolically active, it's not growing, it's not dividing. The endospore really just holds on to the DNA and a few enzymes and ribosomes. It packages just the smallest amount of what's needed and goes dormant. That process of building that spore is called sporulation. When environmental conditions improve, food's available, water's available, that spore will germinate, meaning it will return to a vegetative state. So here in this image, we see Bacillus anthracis, which is the causative agent. When we talk about anthrax poisoning, it's these spores that we're releasing. You can see the bacillus grows as a streptobacillus. We see long chains of bacilli. This entire purple oblong structure would be a cell. So here's a vegetative cell. Every cell would not form a spore all at once in a whole population. Here's a vegetative cell. So it has not yet formed a spore. Here's a vegetative cell. These white circles in here, these are actually the spore forming. This outer part's the spore. Inside here would be the DNA, some ribosomes, and a few enzymes would be inside that. This vegetative cell now, this purple part, is going to die off. It's no longer going to maintain those cellular structures, and we'll end up with just these spores. So here we have a spore. Sporulation is complete on these white circles. Sporulation is in process, where I have this purple cell with a white circle in the middle of it. And I still have metabolically active cells here. So I can see all three stages in this image. If I zoom in on this process, here's my regular normal cell gets to a certain point, it undergoes binary fission, one cell becomes two, and then I grow, I get back to full size. At some point during this life cycle, the environmental conditions go south, there's no more food, or there's a drought or something, and so organisms that are able to will form a spore. And here we see what we're going to do is we are going to make a copy of my DNA. We're going to go through this binary fission process, but we're going to divide unequally. And then we're going to form a spore coat. So this is an organic hard coating on the outside, protein coat that's going to form around it. That's my spore. I have my DNA. I have just the minimal stuff that I need in here. I form that coat, and then eventually this cell lyses, it falls apart. I'm not putting energy in to maintain it, so it's going to fall apart, and I just have a spore. And then when conditions are improved, some water is available, this spore will become metabolically active, start to create structures, and will germinate into a bacterial cell and start the process all over again. Again, take a look at those videos. So endospores are considered a virulence factor because they are able to withstand all sorts of efforts to destroy the cells, and then when conditions are ripe again, are good for growth, out they come. And so they can be present even after we think we've gotten rid of all the pathogens. They're resistant to ordinary cleaning methods and boiling. Really, the only way to destroy them is with autoclaving, so high pressure steam for 20 to 30 minutes, which is quite a lengthy time. There are several bacterial species that can produce these that are pathogens. So anthrax, used for bioterrorism attacks, Clostridium tetani, which causes tetanus, Clostridium perfringens, which causes gangrene, Clostridium botulinum, which causes botulism, Clostridium difficile, which a few of you have mentioned that in class, which is very difficult to kill internal gut infection. So we have lots of different spore-producing bacteria, endospore-producing. One thing I do want to point out, endospores, this is the proper name for a bacterial structure that is dormant. We often abbreviate it and just say bacterial spores, just say spore. Be very careful to double check if you are talking about a bacterial endospore when you see the word spore, or a fungal spore. Fungal spores are reproductive structures, so they're always metabolically active. Bacterial endospores are not metabolically active, but because we abbreviate and just say spore casually, can sometimes confuse them. So make sure you're clear on what type of spore we're talking about. All right, so on to our some more structures. We have the plasma membrane. The plasma membrane, all cells have this. It's set up similarly in all cells. It's a phospholipid bilayer with embedded proteins. We call that the fluid mosaic model. We'll spend some more time on this. The membrane is selectively permeable, and it is what regulates what can go in and out of the cell. So you want to watch the video on diffusion. I am going to post a video lecture on diffusion, osmosis, and membrane transport later in the term. This is going to keep coming up, this idea of diffusion, osmosis, membrane transport. We're going to look at it again when we talk about cell growth and when we talk about control of growth. And you have an activity on this coming up in the next unit. So I'll have that lecture posted to help you through the activity. But these are very common biological concepts that you should be familiar with, and I find students do tend to struggle with those. So facilitated diffusion, we're still talking about diffusion, a passive transport process. We just have a doorway in the plasma membrane. And osmosis is a special case of diffusion where the only molecule we're talking about is where water is going to move through a selectively permeable membrane. So osmosis is a special case of diffusion. What's special about it is we're only talking about the movement of water, and we're talking about that movement through a membrane. Definitely want to watch the videos on diffusion and osmosis that are in here and refresh membrane transport, because this is how things get in and out of cells. So this is a pretty important process, but it should also be familiar to all of you in a 200 level course. Our cell wall on bacteria, we specifically have a cell wall made of peptidoglycan. A cell wall, this is outside of the membrane. It is a rigid structure. Our membrane, that plasma membrane, lets things pass in and out of the cell. It's biologically active. The membrane, let's go back here. My membrane is embedded with proteins. In eukaryotic cells, where I have all those internal membrane-bound organelles, I have separate compartments where all my metabolic reactions, all my chemical reactions that make up metabolism can take place. In bacteria, I just have this one big room. So most of the reactions that take place for respiration, for fermentation, for pretty much all the chemical reactions take place using these membranes, some of these membrane proteins. And some of these will act as doorways to let molecules pass in and out. And some will act as receptor proteins for cell-to-cell communication. A lot of these, many of these, especially in prokaryotes, act as enzymes. They catalyze chemical reactions. So we definitely want to review the plasma membrane and that function. We'll talk about that more, as I said, later. So that membrane is metabolically active. It controls what goes in and out. Our cell wall is not. Our cell wall is not metabolically active. It is the difference. I like to think of it more as a fence around the cell. It is a scaffolding around the cell. It is rigid and provides the shape for the cell. The reason our cells can have those different shapes is because they have this solid wall around them. The cell wall determines shape. It prevents the cell from bursting due to changes in osmotic pressure. So if we are in a very low, a hypotonic environment outside the cell, fresh water, through osmosis, that water would try flooding into the cell, and it would continue to go into the cell, and eventually the cell would burst if it was a eukaryotic cell like ours, an animal cell. But bacteria have that cell wall, so at some point the water will no longer be able to enter. The membrane will press against that wall, and the pressure will be equal to the pressure trying to enter, and that will stop the water coming in. So the cell will not lyse. It will not burst. Similarly, if we're in a hypertonic solution, like a really salty solution out here, the water portion of my cytoplasm would want to leave the cell. For animal cells, that cell would shrivel up. Here, the membrane will pull away from the cell wall. That water will be able to leave, but the cell will still have its external shape because that wall will continue to have its shape. We can define two different types of cell walls in bacteria: gram-positive cell walls and gram-negative. This is going to be our big way to differentiate our first cut in trying to identify a bacteria. We're going to look at the shape of the cell, the arrangement of the cells, and their gram reaction. So here I have cocci, circular cells in clusters, and they're gram-positive. So I can describe these as Staphylococcus. I don't know if that's the genus Staphylococcus because I can have the bacterial cells that stain gram-positive, that are coccus, that are round, and that grow in clusters like this, that are not the genus Staphylococcus. So micrococcus is one, but so I don't know yet based on this, but it would be suggestive. And I can describe use the adjective staphylococcus to describe this. Here I have bacilli. I have to look close. You can see these are fairly small bacilli, so it's sort of hard to see, but these are oblong. I don't see any definite arrangement. So I just see lots. I mean, there are a lot of them together, but they're not in long strips or chains or clusters. They're just sort of all over. But these stain a different color. They stain pink, so these are gram negatives. And you'll talk about the gram staining process in lab. So first, the basic structure of peptidoglycan, which is the primary component of the cell wall. It's a macromolecule that's composed of sugar and short peptide fragments. So peptides, that's the building blocks of proteins. So amino acids. So these are my sugars. I have big long chains of sugars. And these are, don't remember the big long name. There's the big long name, but NAG and NAM. So I have NAGs alternating with NAM. NAG alternating with NAM, and big long chains of these. And then those are connected by amino acids. Amino acids are held together with peptide bonds, so this is called a peptide. Proteins are polypeptides, big long chains. These are short chains, so it's a peptide of the building blocks of proteins, amino acids. And what I tend to see when I look at these repeating layers, lots and lots of layers of NAG and NAM, is a chain link fence. So if I look at gram positives and gram negatives, the big difference is going to be in gram negatives, I have a single layer of chain link fence around my cell. A gram positive has multiple. It's very, very thick. So it would be like having 5 repeating layers of chain link fence around my yard. So this is a gram-positive cell wall, the peptidoglycan, is much thicker because I have lots and lots and lots of layers of it. Whereas in gram-negatives, I just have one or two layers of that peptidoglycan. So gram-positive are structurally simple. I just have lots and lots of layers of peptidoglycan. I have some teichoic acids external to the plasma membrane. You can use these to identify my cell. Gram-negative cells are much more complex. I've got 3 different layers. Where am I here? My peptidoglycan layer is very thin, 1 or 2 layers of peptidoglycan. So very, very thin. For both of these, here's my cytoplasm. So if we look, I'm inside, let's progress from inside the cell, the cytoplasm. Next comes my plasma membrane. And next comes my peptidoglycan, the actual cell wall. And I have these anchors that are specific to my peptidoglycan and gram positives here, but thick layers of peptidoglycan. My gram negative, all right, yep, here I am, cytoplasm. My plasma membrane. And then outside of that plasma membrane, I've got my peptidoglycan, but only a single layer. And then outside of that, I have a second outer membrane. This is a lipopolysaccharide. So polysaccharide, it's a sugar with fats attached to it. I have porins. These are special protein structures through this outer membrane that allow water to pass, because otherwise this is a lipid, water would be repelled. So I need a way for water to get through. I have specific proteins on the outside that I can use to identify this. But lipid A, this is my big one. Lipid A is a component of the gram-negative cell wall that can actually be a toxin. So we're going to talk about this because the gram-negative cell wall actually has a toxin in it. As long as my bacteria is alive, that is not toxic to me. When I kill that bacteria and it's released, then it becomes toxic. And then I connect my membrane to the cell wall through this protein here. So those are my comparisons of a gram-positive and a gram-negative cell wall. Peptidoglycan is the big important one. And peptidoglycan is sugar and peptides. NAG and NAM sugars, repeating long chains of sugars held together with peptides, with short chains of amino acids. There are some bacteria that lack a typical cell wall structure. That means that it's not going to give me a gram-positive or gram-negative stain. Almost all bacteria, I can differentiate gram-positive, gram-negative through the staining process, and I know something about their cell wall. Mycobacterium and Nocardia have an atypical cell wall structure. They really, if I looked at them closely, they would really have a gram-positive cell wall. They have a thick layer of peptidoglycan, but outside of that thick layer of peptidoglycan, they have an outer layer. It's not a membrane, but it's a layer of glycocalyx acids. So these are a waxy substance. And that wax, it works just like wax. If I coat something like wax, it's productive, or it's protective. It's going to protect me from a lot of things. So it's similar to that outer lipopolysaccharide membrane in that way, and that it provides some protection. So it's also a virulence factor. It gives a high degree of resistance. It protects the cell from a lot of chemicals. It also protects it from dyes. So when I go through the gram staining process, this repels the gram stain dyes. So I have to use a special staining procedure to identify Mycobacterium and Nocardia. So what you'll do in lab is the acid-fast staining procedure, because otherwise these would just repel all the stains. You wouldn't see them. I'll just point out, as I said, we aren't going to talk about the two types of bacteria that don't have a cell wall, but do be careful. Mycobacterium looks awful similar to mycoplasma. Mycoplasmas don't have a cell wall at all. Mycobacterium have a cell wall that's similar to gram-positive, but with glycocalyx acids, this waxy outer coating. So I just want to mention that because it is in the book about those mycoplasmas and L-form bacteria that don't have a cell wall. All right, outside of the cell wall, other structures that might be found outside of the cell wall, the glycocalyx. Glycocalyx is a coating of molecules. It's made of a sugary substance, so it's kind of sticky. That's outside the cell wall. It's made of sugars. Sometimes there are proteins or lipids mixed in with those sugars. So typically some kind of glycoprotein. We can have two different types of glycocalyces. We can have a slime layer, which is just very loosely organized. It's very loosely attached. It's easy to wipe off. It can protect the cell from dehydration or the loss of nutrients. So it's just sort of this like outer jelly coating. Or I can have a capsule, which is highly organized and tightly attached to the bacteria. So it makes bacterial colonies mucoid, slightly mucousy, as you can see here, this sort of clearing around the bacteria. That's repelling the stain. We can see the capsule. That's that glycoprotein repelling the capsule, and it's making them stick together. So that capsule lets bacteria stick to each other. Remember, it's a glycoprotein. It's a sugar, sort of sugary sort of thing. So it's sticky, so it can help bacteria stick together. This could be sort of jelly-like. This would be more like Jell-O, more organized and tightly attached. I like to refer to these, because y'all know I'm a Harry Potter freak. I like to refer to the glycocalyx as the invisibility cloak. This is going to be produced outside the bacteria, attached to it. If I am an immune system cell looking to get rid of invaders, I'm going around checking out cells, looking for cells that don't belong. When I touch this cell, when I reach out, say, "What are you?" I see jelly. I say, or Jell-O, either one. I say, "Oh, you're food." Okay. and I don't attack you. So it makes the bacteria invisible to the immune system cells. It also lets these stick together so that I can get a large mass of cells together. Hopefully we're going to come back to the glycocalyx. The glycocalyx, the sticky, the capsule or the slime layer, let bacteria stick together, and so they form biofilms. And a biofilm, you're probably all familiar with, is dental plaque. It's bacteria that stick onto your teeth, and then more bacteria stick onto those because of the capsule, and then more stick onto those. And if we don't floss, we get a nice, heavy, thick coating based on that. Let's see. So glycocalyces can be used for attachment, bacteria to bacteria or bacteria to other surfaces. Fimbriae can also be used for attachment. These are these little thread-like structures extending out past my cell wall or past my capsule if I have one through here. The fimbriae, they're little thread-like fingers. You can see that. They make a fringe. They're small bristle-like protein fibers. And just think if two of these kind of floated by each other, those threads would get tangled. And so these can be used to entangle these cells to other cells. Bacteria that colonize, that cause respiratory diseases, a lot of them have these fimbriae and they get caught up in the cilia in our upper respiratory tract on the cells along the mucous membranes. So we'll look at that, but they can be used for adhesion, so I can stick to other cells. So these are considered a virulence factor because this lets me stick to cells. So if I have fimbriae and I'm going to infect the upper respiratory tract and the person I'm infecting coughs, it's not going to get rid of me. I'm going to be kind of tangled up in there because of my fimbriae. Pilus is the singular, pili is the plural, is a modified fimbriae. It's longer than the fimbriae, and it has a different role than the fimbriae. It can be used for attachment. You'll hear it referred to as a sex pilus often. It's used in conjugation. And I don't like the term mating either because bacteria don't mate. Mating is when you have a male and a female and each contributes half of the genes. Bacteria don't do that. Bacteria will transfer parts of their DNA, right, like the plasmid. And the pilus is used during this process. So here we have the pilus. We have a donor cell that has the pilus, and it's extended it out to this recipient cell. All right, so as I said, science is progressive, and we learn more as we get better tools. So we could see this when I was in school way back a million years ago in the 80s. It was thought that the pilus worked like a straw, that the cell that had one could extend it out, attach to the other cell, and then shoot that plasmid, the copy of the plasmid, over here to this other cell. Turns out now we know it doesn't work that way. It works more like a fishing line, throws it over here, reels that cell in until they're side by side, and then passes that plasmid through the membrane, through the cell wall, through the membrane into the recipient cell. Only gram-negatives have piluses. They are the only ones that can undergo conjugation in this manner. And we'll look more closely at conjugation when we get to the genetics chapter. So this is a really way for bacteria to give the gift of a piece of DNA. So again, watch the video. And all these we're going through fairly quickly. We're going to look more closely at all of these as we go. So that glycocalyx, I mentioned the glycocalyx and how cells can stick together and form a biofilm. So that happens. Here's my surface. This happens on boat bottoms. If any of you are boaters, you have to scrub the bottom of your boat periodically. As I said, dental plaques. If you are not a diligent housekeeper, biofilms will form in the bottom of the sink, in the bottom of the shower drain, on the dog's bowl. You have a surface, some original bacteria settle, the first organisms, and then others come by and stick to each other, and we keep getting more and more layers, which is how dental plaque forms. So these thick living layers are called biofilms. They make the scum in the toilet or shower. They make rocks and streams slippery, plaque on our teeth, and they can form on things like catheters and breast implants and pacemakers. So this is a really big concern in healthcare fields. So the slime layer, or the capsule, my glycocalyx, is really the primary structure we associate with these, but the fimbriae, which allow cells to stick together, can also be involved in that. And there we go, the inside of the toilet, river rocks. This is electron micrograph or a scanning electron micrograph of the lining of a urinary catheter that's been infected with cells through a biofilm. So that's a big way for hospital-acquired for nosocomial infections is through catheters. We also have appendages on our bacteria that are used for motility. The fimbriae have some ability, and the pilus have some ability to twitch. The pilus especially can move a little bit by, twitches. It doesn't work like a flagella. It's not a primary means of motility, but it can move the bacteria slightly. But the primary structure used for motility are the flagella. And so bacteria flagella work like mini propellers. If you remember eukaryotic flagella, they work like a whip. They do a whipping motion back and forth. Bacteria flagella spin. They rotate like a propeller. They're made of 3 parts: the long filament, a hook portion that connects the filament to the anchor, which is called the basal body. That's the anchor. That's the part that goes through the cell wall into the membrane to attach. There's slightly different attachments in a gram-negative versus a gram-positive bacteria, but both have those 3 structures. Bacteria can have different types of flagellar arrangements. I can have no flagella, an atrichous. I can be monotrichous, which is single flagella at one end. Amphitrichous, if you think ambidextrous, I have one at both ends. Lophotrichous is that I have a bunch at one end. And then peritrichous, if you think perimeter, all the way around the outside. The bacteria can use these for motion. They use them for chemotaxis. So they guide bacteria in the direction in response to external stimulus. So chemotaxis, I can navigate toward or away from a chemical. I can navigate toward or away from light, phototaxis. And bacteria can change direction. So depending on which way their flagella is spinning, clockwise or counterclockwise, they will change direction. So if they're rotating their flagella counterclockwise, they tend to swim straight ahead in a straight line, and that's called running. If they want to change direction, they'll rotate the flagella clockwise, and it will cause them to tumble, and that allows them to change directions. All right, so that covers all of the basic structures found in bacterial cells, a little bit about history of bacteria. I know this says study the structures of eukaryotic cells on your own. This is foundational. This should definitely be reviewed. So I'm not going to go through this entire rest of the portion of the PowerPoints. You can do that. I will give you a brief summary overview and tell a little story in the next video about how all of our different endomembrane structures work together: the nucleus, the endoplasmic reticulum, the Golgi apparatus, the transport vesicles, and the outer membrane. So we'll go through that watch the videos, review this, but I'll do a very brief overview of that before we talk more about the eukaryotes of interest to microbiology. So those are the fungus and the protists, my single-celled parasites, and my helminths, my multicellular parasites. So we'll talk about that in the next video. All right. Thank you.